The somatic motor neurons in the spinal cord release action potentials while they move down the axon, causing depolarizations. Then this causes sodium channels to open and therefore allows sodium to flow in. When the threshold potential hits the optimum thresholds, it opens up the activation gates. Sodium comes in, causing depolarizing current. When the positive ions come into the cell becomes more electropositive, and it sends a depolarizing current that moves all across the synaptic bulb. The current reaches a protein that brings calcium into the synaptic bulb to bind to a protein. The synaptic vesicle gets pulled towards the membrane and fuses with the plasma membrane of this axon bulb. Acetylcholine is brought into the synaptic vesicle. Then the vesicle releases acetylcholine and it diffuses across the synaptic cleft and binds onto the nicotinic receptors. Acetylcholine binds to receptor domains, so the receptors open and sodium goes through because it's more concentrated outside the cell. Potassium starts to flow out of the receptor and more sodium comes in compared to potassium going out. This develops more of a positive charge. This change in the charge is called the end plate potential, and it produces a small amount of depolarization. Positive charges move across the membrane to the T-tubule. When the action potential moves down the T-tubule, it stimulates the dihydropyridine receptor bound to another ryanodine protein that's bound to the sarcoplasmic reticulum membrane. Change in voltage, it pulls on the ryanodine receptor, it opens up a hole for the calcium to come out. When the ryanodine receptors are displaced, the calcium goes to leave the sarcoplasmic reticulum. The calcium goes to the sarcoplasm. Calcium binds onto the troponin, it pulls on the tropomyosin out of the way. On the myosin head, the ATP is bound. ATP gets hydrolyzed by breaking the bond of the second and third phosphate, and this releases energy. The ATP attached to the myosin head, it detaches from the actin. When detached, ATP is hydrolyzed. The phosphate is released from all myosin heads. ADP is only attached to the myosin heads. The myosin heads pull on the actin, which pulls the thin filament towards the M line, causing a contraction.